Luminosa, remarkably simple, precisely yours. Luminosa's spiral scan creates an overview in DIC, epi-illumination, or transmission mode, which covers large areas up to several square millimeters in your samples in just a few seconds. This serves as a reference map for further measurements at selected regions or points of interest. Their spatial relationships are saved for analysis. Tiling and stitching of rectangular areas is supported by the Hold Focus feature. Fluorescence lifetime imaging provides new insights into various research areas such as cellular, developmental, or plant biology by offering new experimental capabilities like multiplexing many fluorescent markers, characterizing tissue autofluorescence, removing autofluorescence that spectrally overlaps with marker signals, or sensing changes in the cellular environment. When studying large samples, such as tissue sections, it usually takes time to find a good spot or the feature of interest for detailed imaging. Furthermore, it is time-consuming to manually keep track of how several images from a sample fit together, employing workarounds such as comments or screenshots. Luminosa's spiral scan creates an overview in DIC, epi-illumination, or transmission mode, which covers large areas up to several square millimeters in your samples in just a few seconds. This serves as a global coordinate system for further measurements at selected regions or points of interest. You can select a small region for FLIM, fine focus, and image. In the reference map, spatial relationships are saved for analysis. Save time and simply focus on your samples. Don't lose sight of the big picture, thanks to the overview map. Luminosa, remarkably simple, precisely yours. Luminosa, remarkably simple, precisely yours. Luminosa's sample-free auto-alignment ensures consistent optimal performance for every measurement, even the most challenging ones. Proper microscope alignment is required to obtain high-quality images with good resolution and contrast. It also helps to avoid excessive photo damage when working with live samples. Consistently keeping your microscope aligned improves the reproducibility of your experiments and avoids data misinterpretation. Properly aligning a confocal microscope is a complex multi-step procedure that requires a lot of training. Moreover, it is a time-consuming task that can easily cost a PhD student an hour every morning. If the optical path of a commercial system is not adjustable, users must trust it performs as guaranteed. Luminosa offers not just guaranteed, but optimal performance every day, thanks to its auto-alignment feature. With a click of a button, that procedure takes less than a minute and can be performed even with the sample present in between measurements. This way, Luminosa compensates even for small daily variabilities, increasing reproducibility and long-term stability of the system. The performance of the algorithm is consistently as high as that of our trained experts. As a quality measure for this, we use the molecular brightness determined from FCS measurements. Quality and precision you can trust conveniently increase the quality of your data with a single click. Luminosa, remarkably simple, precisely yours. Luminosa, remarkably simple, precisely yours. Luminosa's CalEx feature measures the excitation laser power and displays it in microwatt without requiring an external power meter. Thus, users can set the laser power reliably in microwatt. High excitation laser powers produce high fluorescence signal, which permits fast image acquisition and increases the signal-to-noise ratio. Yet high laser power may saturate the fluorophores, lead to photobleaching and phototoxicity. This can impair the health of biological samples during live cell imaging and can cause variability in biological results. Therefore, the laser power should be monitored regularly and kept constant between imaging sessions to get reproducible results, as detailed, for example, in this Nature Methods publication. Other commercial microscopes usually state the laser power in percent, but it's not clear in relation to which value. Moreover, if the actual laser power changes, the percentage might stay constant, obscuring the change and hampering reproducibility. To get reliable values, one thus needs to measure the power manually with an external power meter, which is quite time-consuming. It also raises the question where to save that information. Luminosa's CalEx feature greatly simplifies things. It measures the excitation laser power within a few seconds after a single click of a button and displays it in microwatts. This calibration can even be performed between measurements while the sample is present, as no external power meter is required. The result is stored in the metadata. This saves valuable time, increases the reproducibility of your own experiments, and enables a reliable comparison of results obtained with different instruments. Quality and precision you can trust conveniently increase the reproducibility of your experiments with a single click. Luminosa, remarkably simple, precisely yours. Luminosa, remarkably simple, precisely yours. Luminosa's variable PSF feature enables switching between diffraction-limited and larger observation volumes. Luminosa enables you to switch between a diffraction-limited and a larger observation volume in order to increase the observation time window for FCS and single-molecule FRET measurements. The increased burst duration allows observation of slow or hindered diffusion, discrimination of diffusion from triplet states, as well as detection of dynamic transitions between conformational states. The possibility to easily and reproducibly vary the effective detection volume is commercially available for the first time with Luminosa. Previously, this was only possible by manual, careful adjustment of the microscope hardware by a trained physicist, if the parts were accessible at all. An FCS curve is measured with a diffraction-limited volume as usual. With a single click of a button, the observation volume is enlarged automatically. The excitation beam diameter is decreased, underfilling the back aperture of the objective. The tube lens and the confocal pinhole are adjusted accordingly, increasing the effective observation volume. A new FCS curve that is measured inside the same sample looks markedly different. The effective volume is 20 times larger. Thus, 20 times more molecules are inside the volume. The diffusion time is now 6 times longer, yet the value of the diffusion coefficient remains the same. With the large volume, the fit quality is better because it allows a better separation of translational diffusion, triplet state transitions, photoisomerization, and protonation and deprotonation. Quickly and seamlessly changing the observation volume enables you to check for dynamics in single-molecule FRET experiments, observe larger diffusing particles, distinguish different modes of diffusion via spot variation FCS. You can perform spot variation FCS using Luminosa's customized mode to obtain diffusion coefficients without calibrating the volume and to distinguish various modes of diffusion. Advanced flexibility. Unlock new possibilities for cutting-edge single-molecule studies. Luminosa, remarkably simple, precisely yours. Luminosa, remarkably simple, precisely yours. Luminosa can be equipped with both a Flimby Galvo scanner and a piezo objective scanner. You can easily switch between both within a few seconds. For many imaging applications, especially in live samples, a high scan speed is required to observe dynamic processes without motion artifacts. The Flimby Galvo scanner can deliver this. It is even fast enough for confocal single-molecule localization microscopy. Moreover, it's a great choice for scanning FCS measurements due to its linearity. For single-molecule measurements, on the other hand, every photon is precious. Thus, the piezo objective scanner is the better alternative, as it transmits more signal. Most commercial confocal microscopes incorporate Galvo scanners. While they are fast, they lose a few % of the light at each mirror and at the necessary scanning lenses, inevitably accumulating to a signal loss of about 20% depending on the wavelength. This can be a limiting factor in challenging single-molecule measurements, where you need to gather every photon before the molecule bleaches. With Luminosa, you can bypass the Flimby Galvo scanner entirely and use a piezo objective scanner instead to position your focus. This allows you to collect about 20% more signal, depending on the wavelength range. The software suggests whether the bypass should be used, depending on the measurement context. Luminosa is the only confocal microscope that offers this possibility. Additionally, Luminosa can incorporate both PMA hybrid and SPAD detectors. While PMA hybrid detectors can handle higher count rates for faster imaging, SPAD detectors have a higher sensitivity above 500 nanometers and are thus more suitable for single-molecule measurements. Advanced flexibility, high scan speed, or highest sensitivity. Your choice with a single click. Luminosa, remarkably simple, precisely yours.